That's my introductory song. I'm just waiting for it to end so I can show myself. These last few days, it hasn't been letting me do live streaming. I don't know. Maybe it's because I put the link in the playlist. Now I didn't assign it a playlist, so now it is. All right. Introduction's almost over. Then I can go ahead and get to it. I'll go ahead and share my screen as well. I'm trying to fatten up my new dog process. We've been eating lots of very nutritious food. Biscuits, gravies, eggs, pork chops, also pastas, fettuccine alfredo, because I got her ribs when I got her back. We thought it was a forever home, but it wasn't. So 2 years later, at least I got her back. It was someone's second litter, Simon's litter, sister, mate, and now the because Messina is almost a year and she still hasn't had her heat. And this one is 2 years soon, also around June. Messina, and then they're just getting along. They're doing really great. Welcome them in. She's really scared. Now she's loving it. And I had to go ahead and do the Tableau thing on my own. So I just added the Tableau stuff from hold on, is it even sharing it? What the heck? It's supposed to be sharing the screen. I don't see it though, sharing it. Okay, yeah, so I went ahead and uploaded it. That was from our last We're going to look at this intrahepatic cholangiocarcinoma. We got the data already. We got the series matrix information. This is an Epstein-Barr virus associated pathology. They're knocking out the SLC2A1 cholangiocarcinoma, cancer-associated fibroblasts on endothelial sprouting under normal and hypoxic conditions. Oxygen. Cholan has to do with the bile duct, around hepatic liver duct carcinoma, cancer-associated fibro. Expression profiling by high throughput sequencing, not microarray. This is usually bead chip sequencing. Hypoxic endothelial sprouting. We here demonstrate remodeling of the ICC tumor, intrahepatic cholangiocarcinoma tumor. To call that tumor microenvironment as time of a distinct CAF subset, termed tumor-like CAF. We further identify SLC2A1 in TCAFs as a potential key regulator that facilitates endothelial sprouting. Endothelial lining is usually like the lumen of like muscles or the inside, like the endometrium. Endothelial is where the Epstein-Barr virus transforms endothelial cells like squamous cell carcinoma or malignant carcinoma. So it's because you have the cuboidal, you have the columnar, and you just have the basilar. There's also other ones. I'm not an expert on that. I'm just recalling what I can from that. Anything I say, just know it's similar to that. It's in that area, that window of limits. There's definitely going to be some error there. It's not going to be exact, but that's what it is. We further identify, okay, collectively our data provide a molecular basis for the aggressive vascular invasive phenotype of ICC and delineate a tumor, what do we call this? Tumor-like CAF, which are the cancer-associated fibroblasts. So they're saying the endothelial with reduced oxygen, cell signaling axis, TCAF endothelial cell signaling axis, highlighting SLC2A1 as a potential therapeutic target for disrupting tumor angiogenesis in ICC. Blood vessel creation in ICC, blood vessel creation, hypoxia. Further identify this gene, they're saying SL in the tumor cancer-associated fibroblast allows oxygen to the tumor to that region by supplying blood vessels to it. So they want to knock SLC2A1 gene out and prove that the be disrupted, and possibly beyond that, we could assume it stops growing. There are parts of your body that die when the blood supply is cut off. It usually halfway bone right here, the carpal bone. That happens a lot when people fall on their hand. It loses its blood supply, so that it no longer has it, but it's still there. So that's like a dead bone, and it becomes basically a rock, just like a tumor that has new. They never told you that. They never told me that, but it's like, so it just lays there, no longer being supplied, able to contaminate anything. It's not dead unless other things are eating it, but it's usually going to get scar tissue over it, calcified, bony osseous, electrolyte formations in it that it can show a rich osseous material in an X-ray. This calcium rich white, super white on X-ray because they don't have any bone blasts in it. They're not like getting osteoclastic activity. Okay, so that was a similarity comparison of why cutting off blood supply to a tumor is one way of preventing the cancer from spreading. Again, if you have a tumor, if you're healthy enough, you should always get it cut out and anything adjacent to it. That's how our body naturally works anyway, so you should never have it just laying there. But if it's safe and not growing anymore, because your body could eventually find other ways to bring nutrient canals to it, it's just always best to excise it, they say. RNA sequencing analysis of wild type. The reason I say that is because fibroids have a rich blood supply, even though they're capsulated, short zone of transition, and they grow from the inside and out, expanding and pressing on everything else, basically suffocating everything else that's in that region until it gets cut out. Okay, so any tumor you have, just get it cut out as soon as you can. There's nothing that will shrink it or destroy it right away. Unless it's a blood cancer tumor, then there are chemotherapies that do that do destroy it. But if it's a tissue cancer, then chemotherapy is not going to destroy it all the way. So let's see. RNA sequencing analysis of wild-type ICC intrahepatic cholangiocarcinoma, cancer fibroblast cells, and their SH gene. The SH knockout cells under normal, and they're okay. So SH, I don't know what that is. So let's just review what that is. SH in RNA. Short hairpin. So that's a microRNA they're talking about, the short hairpin. So that is something that they use to knock out the short strand of SLCA1 is a gene that is SLCA2A1, but so here's like your RNA strand, right? Your messenger RNA strand, just a solid strand. Well, the microRNA, it's that strand, but then it curves on itself, and then the nucleotides start binding with each other so that it can't go through the ribosome because it's got a bond in the middle. It's usually a hydrogen bond, I believe. What prevents this gene? So they're going to take this, they're going to make them all disrupted and start binding to each other before they can go to the ribosomes, and that will basically prevent them. This gene that they say is causing a blood supply form, and this is a recent study. It is May 11th, and this one's from January 30th of this year, so it's only been a few months. It's from China. There were 12 samples, and that came in the, this is the printout from the page. That came from the Excel file that we just saw. I didn't do anything to it. I just downloaded it. If you go onto the website, you can download the series. That's the information file. And then you can download this GSE 316. That's the series. So that's what we want. And HTTP, just use that one. That's just because that's what I use. That's how I do things. Hypoxic condition. So these are the microRNA, the short hairpin, and it's control. So that's the healthy base. Well, it's not really healthy. It's just the baseline. And then the intrahepatic cholangiocarcinoma, cancer, fibroblast cells. And then we have the hypoxic condition where they take out the gene, the SLC2AC1. And then we have a condition, a control condition. Okay, so here's another control condition. But, so it looks, okay, so we're going to assume these are just the regular cancer cells. These ones are the ones that took out that that have a hypoxic condition. And then these ones, they use the microRNA in the healthy to knock it out, this gene, SLC2A1. And then they have a hypoxic condition for those samples as well. So there, it has not been published, so there's nothing on it yet. We can just check it though by looking again on 16921. We'll go ahead and another new term. Look, and we'll just go here. We will let's just GSC 316. Let's see which. Nothing in the literature yet. Here we have the data sets. Okay. Two conditions. All right. Let's just okay. Hypoxic driven remodeling of the ICC tumor microenvironment prevents emergence of a distinct CIF subset to tumor-like cancer fibroblasts. They identified that gene and in the TCAFs as potential key regulator. I know I just but I'm just trying to understand why there's two different kinds of controls and then two different kinds of hypoxia. And data provided aggressive vascular invasive phenotype of ICC and just a little sprouting, highlighting. Okay. We'll just click on this and then we'll go to it. All right, so here we are, and we're just trying to figure out why there's hypoxic condition. Maybe they're just all related, but they're not, because this is control. This is the microRNA hypoxic. Well, this is the microRNA hypoxic. So this, okay, so we'll have to look in the series. So we'll go ahead and go into our RStudio and look that up. So we will just go into RStudio and then all right, here we are. Okay, environment's empty. This is from something else, so we'll just clear that out. We're going to go ahead and set our working directory as soon as we close out all of this stuff. So we are going to go to desktop, our programs. Let's go ahead and move this now to our app so we can just go there instead of keep going in, because the further in you go, sometimes it won't recognize that folder anymore on Microsoft. Okay, set this as our working directory. Go over here, set as a working directory. All right, now we're going to go ahead and make a new markdown file. This one's going to be intrahepatic cholangiocarcinoma ICC C316921. All right, analysis and ML. We'll just leave it like that. Okay. Wow, it's making me ask. Okay. Okay, so I do that. Now I'm just going to go ahead and save it again. Intrahepatic carcinoma ICC 16921. Analysis and ML. Okay. Now we'll just go ahead and remove all this. Let's go ahead and make some chunks. Let's just go to 200. And we'll just Control S save. Let's write a little brief description. Intrahepatic cholangiocarcinoma, or ICC, in GSE 316921. Study about the knockout of a gene that provides angiogenesis to the tumor microenvironment. No published date, but study is complete January this year. In it, the description says the hypoxia in the ICC tumor microenvironment creates tumor-like cancer fibroblasts, and then it also looks at the short hairpin. I take that to be a microRNA to inhibit this gene, SLC2A1 as SLC2A1. We saw in the samples there seemed to be 2 sets of control and hypoxia, but in one set there is no SLC2A1, and in the other set, confused on what is the control for the hypoxia that led to the SLC2A1 gene as the target of create hypoxia. If there is a control with hypoxia already, it did say something about or similar to copy number variant. Maybe we can discover something here to explain more in the data provided. In 12 samples with 3 of each class in 2 sets. So there was already a data table provided. Series to explore. Okay, let's control a save. Now we're going to go ahead and go to our first chunk. Let's explore this data first. Okay, so let's actually read it in our libraries first. So you should have these installed already. Install dot packages, parentheses, quotation mark, name of the package, and quotation mark in parentheses, play. Wait for it to install. Then you're going to go to a library as a function, and you're going to use our markdown. That's all we're going to really use right now because I just like the way that it puts the table in the knitter package. And now we're going to go ahead and look at this. I'm going to move this over here. Okay. We're going to open that data. We'll just call it data. We're going to read read.csv this file. It is called GSE 316921 processed data.csv. We'll just see what it says first. Let's look at it in the object window first. So it looks like the row names are X. That's the gene name. Actually, it had row names. And now we're just going to rename that first column. So this is the cancer fibroblast, SH, and then it's the SLC. And then it says hypoxia 3. Oh, good. It has the names of it. So now we already have the sample. We couldn't anyways because they didn't have any samples. This is what they provided. Oh, there were samples. I didn't upload each one individually, though, because they already did this here. So I'm going to go into it. And then let's just look at the, we'll just look at it in the knitter table. So we'll go ahead, even though we already looked at it, we'll just look at the first 10 rows. We'll just run that line. And then we can look at the column. Let's go ahead and we'll just put the dimensions data just so we see it. I mean, because when it knits, you know. Let's look at the column names. Really, I don't like this hover feature. Okay, so we're gonna name names of data. The first, we're gonna name it Gene. We'll just name it Gene. Okay, and then we're gonna go back and see if it works. Okay, good. Now, like the work is, there are F for cancer fibroblasts, the controls of then the hypoxia, and another set of controls, but for the short hair and of the targeted gene, and the hypoxia of those controls. I think now that the hypoxia is something they do in the lab to these samples to see what happens to the controls when they remove oxygen. To see, as the description says, what happens with the endothelial tissue. Okay, so now let's look at the information on the details. Okay, so let's go ahead and read in that. Oops. I didn't look at it in a notepad. That would be helpful. Not. Oh no, it will. Only one variable because it didn't split it on this. So actually, this one seems like it's already pretty good. See, let's just read dot table actually, because it looks tab delimited. Oh, line 24 did not have two elements, so we're going to in row read in lines and see if it works. We'll read in 22 lines. Okay, good. So let's call this series 22 lines and run it. Let's remove in our console to not confuse us. Okay, good. Now let's go ahead and look at that. We'll highlight that and run the selected line. Okay, great. So let's go ahead and lower this console because it's too high up. Okay, we're gonna look at what we have here. You have talks about the knockdown and intrahepatic cholangiocarcinoma, cancer-associated fibroblasts on endothelial sprouting under normal and hypoxic conditions. Okay, great. Looking at the endothelial sprouting ICC genes of the cancer fibroblasts, and then whenever they reduce the oxygen to it, when they suffocate it. And it has some information here. Hypoxia is a critical determinant of endothelial sprouting. We here demonstrate the hypoxia-driven remodeling of the ICC tumor environment from the emergence of a distinct CAF subset, termed tumor-like CAF, TCAF. We further identify SLC2A1 and TCAFs as potential key regulators of endothelial sprouting. Although the underlying molecular mechanisms remain to be elucidated, collectively, our data provide a molecular basis for the aggressive vascular invasive phenotype of ICC and delineate hypoxia TCAF endothelial cell signaling axis, highlighting as a potential therapy. And that was in the description. It's good to have it there. Sometimes you just over again, and sometimes the words make sense. Okay. Sometimes you have to look up other words to make sure you're understanding it properly. So RNA sequencing analysis of wild type A1 knockdown cells under normal and hypoxic conditions. Okay, so let's look at what that is. That is the overall design. This is the status. Let's get, it was the, okay, so this is the overall design. The one above it is the summary. And the type, okay, that's a type of sample profiling, series type. Next page. Okay, information if you want to contact them. I've never contacted anyone. Okay, and then let's go ahead and see what happens after we skip 22 lines too. So we'll call this series 22 skipped. Read dot table. GSE 316921 series matrix.txt. And we're going to skip 22. Skip equals 22. Line 44 did not contain elements. Okay. So we're going to skip 22, but we're going to go ahead and read in 20. After 22, there's 22 left. So we'll just go ahead and read in. We'll start with 22 just to see. All right, let's see if it's different. Highlight that, run the selected line. So we have biliary tract tumor tissue. That's important. That'll go in our summary. Let's just write this here above that we did say that it's doing. Details above. The data does seem to suggest this is sort of exactly what happened in the what's that name of the series summary and series overall design. In the series summary and series overall design information. Let's see what else. Series data to help us understand understand what we are working with. And here in the above, the tissue type is biliary tract tumor tissue taken. Is ICCCAF cells 1. Here. Oh, control condition 2. Okay, so this is telling you what sample ID it is under each one. So biliary tract tissue, tumor tissue taken. Okay, let's see if that's the truth for all of them. It seems like all of them are biliary tract tumor tissue. So they're all from biliary tract tumors. Okay. Taken for all samples of control or SHSLC2A1 conditions, hypoxia or not. Okay, that's great. All right, let's see if there's anything else. Use trim galore describes what they are doing if you have these tools here and you want to reproduce it. It's a fibroblast. That's what builds fibroblasts. Let's look to see if that's the same one. And they are all, and it does say the wild type. So if you go to the wild type, okay, so the first sample 6 are all the wild types. Okay, these are the ones that they took out the oxygen. Okay. So the wild type total RNA of the biliary tract tumor tissue, they're all fibroblasts. Okay. They are all blasts. The first samples are WT for wild type. That's what I read before. It's the same. Assumed. Okay, so okay. Let's see if that changes. Looks like it's all the same, like this middle stuff so far. All right. 6 to a 1 knockdown or inhibition. Okay. Let's just make sure, because we're looking right here, genotype. These are the controls. And these are the hypoxia. Okay. So now we know what we're working on. The first 3 samples are controls, WT, then hypoxia, WT. Next 3 samples, and following 3 samples are the SLC2A1 knockdown, and last 3 following that are the S1 knockdown with so now we can look at the data, explore it. All right, let's just look at it in here real quick. Okay, so we're looking at the controls right here. There's some variation within the controls. Wild type. Then we have the hypoxia version variation. Some fire. We'll just look at we're just gonna do the fold change of these. So we're gonna get the means. We're gonna get the baseline controls in each set. And then we're gonna get the fold change of this. Okay. Let's control S2. So now let's just, yeah, okay. So data by healthy, not healthy, baseline. Okay, let's do wild type. WT. WT mean. Okay. WT no hypoxia mean. Rho means. Rho means. It is rho means. Rho means data. It's going to be all the rows. Data by WT hypoxia mean. Rho means data. See, it's going to be rows 5, 6, 7. Data by SHSLC to A1. No hypoxia mean. Data. It's going to be rows 8, 9, and 10. And then data by SHS data. See, I mean, it's going to be columns. I've been saying rows, right? It's columns, 11, 12, and 13. Let's go ahead and run that, and then we'll just look at the column names. So we have our mean values. Let's just make sure that they look good. Okay. Now looks like it's already been like normalized here. So I think when they put the stuff up there, they already normalize it. It will also tell you in the series information. I've never had to normalize any of this data because I just assume it is already. Okay. Well, series, that's not even, we don't even have that one anymore. Okay. What? FastQ is used to read all the raw data from quality control. Quality control includes the normalization and filtering. And they use something to do the quality control. Just verifying it. That's fine. Good. So now we're going to add the fold change. These two sets of genes looking at hypoxia in both, but one with the inhibition of SLC2A1. I'm going to call it inhibition because that's to me what knock down or knockout means. It's all semantics, as they say. So let's go ahead and add the fold change values. Data by, let's go the last ones. Okay, no, we're making a new one. Okay, data by wild type. Okay, so we're going to do data by wild. Okay, so it's going to be wild type hypoxia. That's going to be the change that they made to the no hypoxia. That's the baseline no hypoxia, okay? So we're going to do over no hypoxia for that. And then the other fold change is going to be SHSLC2A1 fold change data by SHSLC2A1 hypoxia over data by SH no hypoxia. And then we're going to okay, great. Now let's go ahead and we'll just look at it as a table. So we'll go ahead and do page table data and just the first 10 rows. All right. It says there's 19 columns. And I'm just going to go ahead and put here. It is 36,242 by 19 in dimensions. Okay. All right, let's Control C. Now, let's go ahead and get the top genes. Let's look at this. Let's see what happens. Let's go ahead and order these genes. Let's order the genes by the wild type fold change and get top stimulated and top inhibited genes of 10 each. Okay. Data WT ordered. FC ordered. Okay. Data order. Data by wild type fold change decreasing equals true. We're going to keep all the columns. And then let's look at page table data. WTC ordered first 10. But let's we have to make a vector 1 through 10 and 36 to 43 to 33 to 36 to 42. All the let's go ahead and do that. Okay, so genes, those are the down inhibited. You know what? We might have to get rid of the zeros. Oh, I feel like there might be some some nans as well. Okay, yeah. So there's some infinites and there's some nans. So we're going to have to get rid of those. Let's remove the NANs and the okay. So and do that. data removed name. Okay. Okay, data. Let's see. Ordered. Or like not is dot NA. Data ordered line. All top field change. There, let's see if it works. All right, good. So there were a lot of NAs. The dimension is 21,000. Okay, so we'll use data removed NaN not is dot infinite data removed n full change and all those. Now let's run this line of code. And less, even more less, even less variables. Wait, 20,000. 178. Okay, great. So now let's look at this. We might have to still remove the 0. So I'm going to go ahead. Let's just look at this one, okay? Let's look at it. Let's see. We can look in our viewer, and we can see this is our targeted table. There's a lot of zeros, and really what happens is you just get all of these that are just alphabetized. So let's go ahead and just remove all this, make it so that everything's greater than 0. Data clean to wild type FC. I'll just put it like that. Wild type FC. Let's put a underscore there. Data clean wild type FC. So we'll go ahead and use this data removed in NANs, and then we're going to take which data removed in NANs by multi-fold change greater 0. And let's run this line of code. And we have even less, 18,474. Okay, so let's put and remove all 0 values for genes. Okay, let's Control S save, and let's look at this. This is where we're at. Okay, so looks ordered. All right. So we'll go ahead and go over for this. Okay. Let's write this out because I feel like we could use this later. .csv data clean. Okay. Row.names equal false. Header equals true. Oh, let's make that on a new line too. because I want to not run it every time. Let's just go ahead and cut this one out real quick. Oh, right. I have to name it. Okay, let's cut it here anyways and put it on the next chunk. And then we're going to put eval equals false because we're just going to run it here and we don't want it to keep overwriting it. Okay, and we're going to name this data cleaned wild type FCs 474 19.csv, and then now we'll write it out. Okay, great. So now we're going to go ahead and take the top genes from this group of wild type controls and hypoxia, hypoxic environments. Top wild type. Oops. Let's put 20. We're going to go ahead and take data, clean this one, and it's going to be the first 10 and then the last 10. 18,465 to 18,474. Okay, and then we'll go ahead and look at what we have. Oh. I'm just getting used to this dark version. All right. So these are stimulated ones. Stimulated genes. Right here. This is where we're interested in. The top. By mean anyways. There's only 3 samples each, and we can see that there are some zeros right here for this gene, but yeah, I didn't want oxided in the control. Okay. Doesn't, oh, my leg just fell asleep. Oh yeah, we didn't even use those other ones. There were zeros, but let's see here. These samples do have zeros in the control. There's only 3 samples each too, so I seem to make it in the all right, we'll just do that because that's what we've been doing. There's other ways to play around with things. Or whenever you're removing the nans and the zeros, you can take the median. That's one way because then it shows you is in the I didn't use this one. This is so that's not relevant to this group. We use these ones as controls. Wait, they have controls in the other group. We use the one SHN. And C. All right, I'm just looking at these names real quick. Okay. Okay, so it does have SLC. All right, just making sure, like they were saying that they were mixed, because the SH. Not what it goes for. I mean, you would think that it would just say wild type on it. That's what it says in the series data. Same thing. Top WT20. Just have that there. Let's go ahead and erase from our I guess source environment object. All right. So now let's do the same. Get the cleaned up data of new in and there's greater than 0. Full change values of the SHSLC2A1 group of controls in hypoxic condition. So we're going to go ahead and do data. We'll just put SLC ordered first. Do it that way. Data to just order it by that one, that fold change. Hairpin SLC2A1. Decreasing. Oh, let's put order. We didn't put the order function. Decreasing equals true, comma for columns. Yeah, so let's just see if there's any infinite in this one. Run selected line. Okay, SLC ordered. We'll go to that. We'll go to that column. Yep, we see right away there are some infinites. There's most likely some nans. Rid of the SLC ordered. Not is .na data SLC ordered by this one. Okay, all the columns, and then data C no inf data LC no man data SLC no and by we have to okay SHSLC2A1 change. Okay. What is the infinite? Okay. Let's run these lines of code. What this one is in dimensions. SLC no nans 22461 by 19. And then this one is 20 zeros. Data. Okay. SLC no inf nan. Okay. And then data. This one by this one greater than 0. Ooh, we need to make that a which function. And then let's go ahead and run that one. All right, and this one has Data, where is that? SLC clean 19,100 to 519. All right, let's go ahead and write that out. Let's make a chunk. Measure eval equals false because we can just run it right here. We don't want it to run it every time. Okay, and we'll just write this to CSV. Write down CSV data full change 19,102 by 19. That's CSV row dot names equal false. So we'll write that out. Okay, now we're going to get our top. Of 10 stimulated and 10 inhibited. Let's go ahead and look at this table too, though, first. Just make sure everything is ordered. Let's run that. Oh, whole table though. Just like the Actually, let's not look at that because we can't really see unless we look at it in here. This is the one. Oh yeah. Hold on. No, this is the one right here. And we just want to make sure everything down and not is good. No zeros. Awesome. Very tiny numbers, but not zero. Okay, good. Now we're gonna get the top genes of this data. Top SLC 20. Data SLC clean. The first 10 and the last 10. So it's gonna be 19, 19, 0, 0, 91 to 19,102. Let's see what that does. Oh, 93 is what I wanted. Right. All right. They're playing. They're just right now. All right. Let's see. So. It looks top 10 stimulated inhibited ones. We are going to next. Now we have our top of both groups. Let's see how these genes play out. And first, classifier to predict the class of the sample. Let's take a break real quick. Because I want to put them in the bathroom. Hey, I'll be quiet. So now we're going to do some machine learning. Let's see how long it's been. It was like a 5-minute break right there. Long. Let's go on back to the line for the machine learning. I'll just run both of them again. And then let's just put in here the chunk equals false. And let's just run it again. Okay, good. Control S, save. Go to the bottom. Okay, good. So let's just look at our genes first. So these are the top 20 wild type genes in hypoxia compared to IACC. Top WT20 by gene. Okay. And these are the top 20 genes in hypoxia, non-hypoxic condition, knockout inhibition of the gene SLC2A1. Okay. All right. So our regular, we should have possibly seen a change with that gene. Just doing what we were doing, but I mean, it's not necessary because they, when they inhibit stuff, it doesn't completely knock it out in some cases, like our last. There was something that was study and it was not completely knocked out. Okay, so let's do some machine learning to see if this can predict the class relative to the data set the top genes were in. There are 3 samples of each. We will train on 2 samples and test on the remaining on 4 samples, but 2 each on the remaining 2 in each group or data set. For the wild type, let's create our matrix. Okay, so type, well, let's just call it DF. Okay, we're just gonna use top WE20, but we're gonna use all the rows, but only the first feature. Okay. Class, let's make that right now. It's just going to be because they're in order already, so it's going to be ICC first 3, no hypoxia. The next one is ICC hypoxia. Let's just make sure that we have the right, we typed it out right. Good, there's 3 in each. That's our class. Make our matrix. So data.frame, transfer function, WTDF. And then we're going to only use the 2nd through 7th columns. And okay, I believe that's it. And then the column names, W, T, and X, are going to be the genes. W, T, D, F by gene. And then WTMX by class as dot factor class. Let's look at our table. And we have the genes on top. There's 20 genes, and there's only 6 samples. First 3 are the ICC. The next 3 are the ICC with hypoxia. All right, and we can check hypoxia are the last 3. Controls. No hypoxia the first 3. Now let's group our training and testing data. So we'll just do this manually. Let's set our seed to so it always reproduces the same results using the random key generator. Training set equals WTMX, and it's going to be by row. It'll be the first 2 of those, and then the next 2. And we'll just use the last 2 of the last. So, and then 5 through 6. So let's just do 1, 2, 5, 6. And then, of course, all of the rows. And then the testing will be the ones we didn't use. 3 and 4. Okay. And let's just make sure we have the same amount of samples in each to train on. Good. And then the same for the testing set. Good. So now we're going to go ahead and make our random forest model. It's going to be training, and then the features are okay. So what is the for the columns, because there's how many? Let's look at this. WT MX, there's 21 variables. It will be 1 through 19, and then training by class. Mtry equals 1/3 of 20, so it's about 6. That's usually what they say to use. And then the entries, let's use 5,000. Confusion equals true. And then the next one is importance equals true. And then we'll go ahead and do to compare. All right, look at that. The class error is, that means it predicted with 100% accuracy. That is good. The genes are good for this training set on class prediction. The 2 ICC classes samples were predicted correctly, as well as the 2 hypoxic version samples. See. Let's see how well it did this model on the hold out validation testing sets. Predicted wild type predicts RF is a model on the testing set. Let's make our table results. WT data.frame predicted equals predicted WT underscore WT, and then actual equal testing by class results WT. Let's go and run that. Great. It looks this model was good with this set of genes in determining the class of the sample. Let's see the importance of these features. RF wild type by importance. So for all of these genes, we can see the importance and the weights that they used. All of them are in the thousands. So whichever is the highest, this looks like the highest one for the ICC group, and the highest one for, and the most weight in it not being an ICC would be this one. Also the same for the ICC hypoxia. And they seem to be pretty much the same across, probably because of the few samples as well. I mean, all right. And then, let's go ahead and do the same. So these wild type genes, of 20 of them, are predicting the class of that subset of ICC in hypoxic conditions. The importance of the features was identical for each sample. Could be due to the small amounts of samples, or something else. Let's now make a matrix of the knockout SLC2A1 top 20 genes and test those out in its subset. Okay, so this one will be SLC. I'll just do SLCDF, and we're just going to use, and it's going to be the first, the 8th through 13th, and 11, 12, 13, because that's the last 6 columns for those samples. And our class is still going to be the same arrangement, but it will be with, let's just make sure just to verify. Looks like the hypoxic are always the last 3 samples right here. Oh, actually, those are our added. Yeah, so the hypoxic samples are at the end, and the baseline is right there. So they knocked out this gene, and then they also had a condition where they knocked it out, but also had hypoxia. So that's correct. And now go ahead and name a class. Feature, it's just gonna override that. We'll just call this class 1. Okay, and it will be ICC SH, SLC to A. We'll just copy that twice and paste it twice. And then the next one will be ICC SH SLC to hypoxia. And we'll copy and paste that twice. We'll just copy it. Okay. So we'll go ahead. Look at it. Very good. So that's what we want. The controls and the hypoxia at the end. And we have the gene in the first column. Now we're going to go ahead and make our SLC matrix. Data.frame T function SLC but we only want to use 1 through 7. Wait, 2 through 7. Wait, 2 through, 7. Okay. Right? Okay, because it is still 7 variables, so, and we want the gene separate to have that in there. 1, 2, okay. And then let's call the column names. Let's name them. That's by this, we'll use this frame gene name. I'll add SLCMX by class as dot vector class 1. Now let's go ahead and look at the matrix table. Oh, okay. Looks good. We got our controls at the top, hypoxia at the bottom. Let's make sure our samples are the same. Good. That's how it is for the samples. Now we're going to go ahead and make our training and testing sets. You can put whatever number, but make sure it doesn't matter really. Just, whatever. You can put whatever number there. It does matter though if you want the same results, you have to put that. But just to be safe, just go ahead and do it. Set is going to be the SLC. And we're going to manually pick on by row. So it'll be 1, 2, and 5, 6. The first 2 and the last. All the rows. Testing. The third one and the fourth one, the middle ones. Just because let's make sure there are 2 because there's only 3 samples each. We just want of the class feature. Good. And now the same for the testing feature. Good. 1 each. That's right. That's the remaining. So now we'll build our random forest. Okay. Our FSLC random forest training 1 through 19 for the features, and we're targeting the class feature, training by class. Genes will tree in tree confusion equals true. Equals true. Let's look at the confusion matrix. Good. These genes are also in the class of sample has 100% accuracy for both samples in this group. About the testing set of holdout samples. Okay. See. RF on the testing set results 2. We'll just put 2. Let's just put 1. Data dot frame predicted equals predicted result actual equals testing by results 1. Good. All right, go right. %, accurate. And predicting. So for this study, it looks like we could see that these things by whole change comparison in hypoxic states of both groups, wild type intrahepatic cholangiocarcinoma and the inhibition of the SLC2A1 gene in a separate group of hypoxic state comparison should the study and change occurred that was demonstrated change in these genes for the hypoxic state. Let's see if we look at what these genes are. By merging them with a separate file that has a brief description of their some control essay. Okay, so I do have the ensemble gene package to merge my ensemble, but recently we had another file that had a summary. Maybe not. Okay, I thought I saw someone, but okay. So let's make a path. We're gonna say eval equals true, but include equals false, so we don't show path. And we'll just copy that same path. We're gonna have to go get that file though. And it is in all pathologies. ensemble, and we're just going to copy, right click, copy the path. We'll go back here. Oh wait, actually, okay. Type this and switches back forward slashes for forward slashes. Over here, select in selection, put a backward slash, and then a forward slash in replace. I'll make sure in selection is selected. Good, 7 occurrences. That seems right. Now, I'm leaving this here, but I'm not going to run this yet. Let's go ahead and do this one. Addition to your ensemble ID data set to merge. Okay. And we're going to put eval equals false. Okay. And then we're going to go ahead and go to the next chunk. Set our working directory, set WD function to path. It's just only going to set it in the chunk. We're going to have to read it in first, ensemble reading.csv, and it's going to be this right here. Let's go ahead and cut it right here. And we'll do quotation marks. Let's see. And then here's table on, and then just the first 10 rows. Oh, we didn't run it yet. Okay, hold on real quick. We have to run this first. Let's close this microscope. Okay. And then now we can set the working directory. Okay, so these we're going to merge by the gene name. We'll get the gene ensemble ID, but I just want a description, at least the name, you know. And it also has the chromosome location. We only need the first 4. We don't even need the first 4. I just wanted that little information on what the name is. We don't actually have the summary because we're going to have to go get it at a separate time. But at least we'll know what it is, okay? So we'll go ahead and merge this. We might as well just keep the ensemble ID as well. So we ran out of chunks, so let's go ahead and make a few more so we can do this. I mean, we don't need that many, but okay, let's go ahead and merge this with the top genes of each group, but only the first 3. Wait, first 3 columns? Let me make sure. Ensemble. I'll just do the first 3. Add these to our only the first 3 columns to our pathology database. It would be nice to explore these B genes as well, of course, since we do want to connect this with the differences in those genes. If they're there. Okay. All right. So, okay. In the first 3 columns. Okay, so we'll have the top 20 merged. With the wild type. And merge ensemble and top WT20 phi dot x equals gene underscore name all lowercase phi dot y equals gene capital G. Look at this. Okay, so let's look at the description. These are the stimulated ones. Aldehyde dehydrogenase 1 family member A1, anactamin 3, capping protein regulator myosin 1 linker 3, CD300A molecule, cell death inducing DFFA like effector A, CXC motif chemokine receptor 4, iodothyronine deiodinase 2, Figetin microtubule severing factor, guanylate binding protein family member 6, 4-hydroxyphenylpyruvate dioxygenase. Now the inhibited ones in the wild type, when a hypoxic state, these ones were decreased insulin-like growth factor binding protein 3, inhibin subunit beta B, long enterogenic non-protein coding RNA 520, MAS related GPR family member X3, Notch 2 N-terminal like A, P10, P1 antisense RNA, S100 calcium binding protein P, succinate receptor 1. Now the stimulated ones had a chemokine receptor 4, a motif, a cell death inducing the hypoxia. This is the hypoxic. You know what hypoxia does. These genes are stimulated. And it says that the endothelial outgrowth or something like that. With a tumor-like calcium fibroblasts. Okay. Now let's compare it with what happens in the knockout one, top genes. Let's now merge ensemble descriptors with the top 20 genes of knocking out SLC2A1 with SH SLC2A1 in hypoxic state. Okay, so for this, let's go and go top SLC merge top SLC. Let's do ensemble. Top SLC20 phi dot x equals gene underscore name all lowercase phi dot y equals gene lowercase g page table top SLC merged. Okay, so now let's look at what these genes that are stimulated do in the hypoxic state whenever they knock out the SLC2A1 gene. These are stimulated at ADAM, metallopeptidase with thrombospondin type 1 motif 18, anoctamin 3, Acate scoot family BHLH transcription factor 2, fatty acid binding protein 5, pseudogene 3, glucosaminosyl N-acetyl transferase 3, mucin type, glutamate ionotropic receptor NMDA type subunit 2A, insulin-like growth factor binding protein 1, inhibin subunit beta B, INCA2 antisense RNA 1, inositol hexaca phosphate kinase 3. Now let's go to the inhibited ones. Janus kinase and microtubule interacting protein. Sounds like a signaling network. KISS1, leucine-rich repeat neuronal 3, leucine-rich repeat transmembrane neuronal 1, mitochondrial coiled coil domain 1, These ones are all inhibited in the hypoxic state when they knock out the SLC2A1 gene, as well as POM121 transmembrane nucleoporin like 9 pseudogene, ribosomal protein L10 pseudogene 6. Ribosomal protein is something that makes the ribosomes to make proteins, but it's a pseudogene. Ribosomal protein L7A pseudogene 50, small nucleolar RNA host gene 21. Those are all inhibited. All right, we'll have to find out like a description of what those do. And so let's write these out. Just because. In fact, why don't we just go ahead and add a column to each of these that says that they are what they are: top fold change wild type, top 20 wild type. Okay, so we'll do that. Top 20 WT merged by source. Oh, not source. Whoops, is it like data set source? Okay, we're gonna say it is top 20 genes by fold change wild type hypoxia in ICC. Okay, and then top 20 top SLC merged by data set source, top 20 genes by fold change, SH SLC to a 1 inhibition knockout in hypoxic state ICC. Okay, now let's just combine these with the robine. Top genes ICC. Okay, arbine. Top 20 WT merged. Top SLC merged. And then let's get and look at it. All right. And we're going to go ahead and write this out. It's So I went ahead and added those. I guess I didn't select the proper way for these. So I'm gonna have to go back. Good thing I caught that. I'm always catching stuff there like that out because I'm always looking at it. Just trust the code from repeating it. Okay, so what I think I'm doing ensemble, but we only want the first 3 features. Okay, that's what we said, right? Okay. Wait, do we want the location too? That would be nice too, actually. 1, 2, 3, 4. Actually, the 4th. First 4. I didn't scroll through all of it before. Now it's good. 22 columns. That's better. Let's do the same for this, for the other one above it, because that was the last one. These are the wild type. 22 columns also good. And these are the right samples. Good. I mean, actually, they're all going to be there. All the samples are there. Okay. So now we're going to go ahead and write this table that we just combined. we got to go ahead and run this chunk too, or else we're going to write out the wrong one. All right, and that one has 23 columns because we added the data set source. Okay, good. So now we'll write this out. Let's put eval equals false so we don't write it up every time and possibly have some errors. Oh, let me do that. Okay. That we don't want. So just write this one time. This one is called, what is this one called? It's temporarily named Top Genes ICC. Okay, good. I'll just put it in the Top Genes ICC. Top Genes ICC. Okay, I'll just go ahead and put the sample number 2316121.csv. Names equal false. Let's write that out. All right, great. Next time we will have to add in some gene summaries to see these genes we're doing and add these genes to the pathology database because they are good predictors for the class, the class of the sample in their respective subsets. Let's quickly return to the larger cleaned data to see if a few EBV genes are present. Some are LMP1, LMP2, PDL1, and PDL2, but the different names. Get CD74 and PDLG2, I think it was. Let's look real quick. I believe it was this one. All right, well, okay, so we need to go up even further. And the study I'm looking for was the Hodgkin's, because there is an alternate name for that one, and the diffuse large cell lymphoma. And it's going to be in the actual study. Let's see where it's at. See. PDCD1 LG2 and CD274. Okay. PDCD1 and CD274 right here. CD274 and right here PDCD1 LG2. Okay, cool. I'm gonna go ahead and close that one out. Let's see how long. We've been here 2 hours. We did quite a bit to this because it's not late yet. Okay. EBV, we'll just call this EBV genes. We'll call it LMP1. LMP2, PDL1. Oh, actually, CD274 and PDCG1LG2. All right, let's run that, make a vector. Okay. Now let's see at our environment, the names of the cleaned one, data clean. Okay. EBV WT. Okay. Data clean. Data clean by gene percent in percent and let's look if we see anything. DWT. Oh, two of them are. Okay. Looks like PDL1 and PDL2 are here. PDCD1LG2 are in the data set from above in wild type cleaned of infinites nans and greater than 0 for full change. Let's see. PDL2 is on the top. Let's get the full chain. This is the wild type one, so they're both inhibited. They are both inhibited in the hypoxic state of wild type ICC. Now for the cleaned data of hypoxic state in ICC with the knockout of SLC2A1. Okay, so this will be EBV SLC. Okay, but it's data SLC clean. Which data SLC clean by gene percent in percent EBV. Page table EBV SLC. Okay, both of the same PDL1 and PDL2 genes are also in the ICC hypoxic subset group of knockout SLC. SLC. Oh, now I'm going blank sign. All right, hold on. SLC. What is it? SLC 2-1A? 2-1. Wait, 2A? Hold on. SLC 2A1. Gosh. A1. Okay. Let's see if it's still inhibited. Wow. Also, both inhibited. PDL are chromosome 9 genes. All right. So we're going to just take a couple of these data sets and we're going to go ahead and just add the whole data right here. Yeah, because we'll just add it and then we're not going to have it cleaned whenever. We'll make a folder of it. Okay, cool. Next time we will explore the gene size for each of these top 40 genes in ICC hypox states which site and knockout SLC2A1 with S8 short hairpin SLC2A1. Okay. All right, control is safe. Let's go ahead and knit this. I'm gonna have to log in, but you won't be able to see my password. My thing just charged it. Okay. Okay, we'll publish this. Let's just look at it and see how it looks. All right, let's publish. I've never used any of the other ones. All right. Okay. So this one is let's just narrow this a little bit. Okay. All right. Exploratory data analysis without plotting, just reading the series text information and then use full change data of the supplied data set on intrahepatic carcinoma or ICC. Two groups to compare hypoxia in ICC wild type and in ICC with a knockout inhibition of short hairpin or SLC2A1. We then separated the groups after getting fold change value for top genes stimulated and inhibited without infinite or zeros. and used a quick random forest classifier to tell if the genes would be good in predicting the class of the group. And 100% accuracy in all, in both groups for training and testing. Small set of samples. We then looked at the genes, recent studies in our projects to compare LMP1, LMP2, PDL1 or C4, and PDL2 or PDCD1LG2. Only the PDL1 and PDL2 genes were included and both inhibited in both groups. All right, cool. So we'll go ahead and I'll get this link and share it on the details. ICC GSD 316921 analysis ML random forest EV PDL1 PDL2. Okay, cool. Let's get that link and I'll share it. I'll put it in the details. Here it is. I'll just Control C copy. I'll go ahead and all right. Intrahepatic cholangiocarcinoma or ICC. Good analysis results. Let's mention one name. And EVV comparison of full change. All right. We'll save that. And then I feel like there's something else I need to do. Close that out. It's already published. Yes, let's go to the folder. We can close this out. It's fine. It's already saved as that RMarkdown file. It's just in our studio environments. So we're gonna go to this folder we were working in, and then we're going to make a new folder, and it's called intrahepatic cholangiocarcinoma ICC datasets 16921. Okay, now we're going to copy. Oh, actually, we didn't write out the whole data, huh? Okay, hold on. This was so short, I can just rerun it again. So we'll just rerun everything. Open it up from the R Markdown file. Let's just run all. And I'm going to write the data to the CSV because we only did the cleaned ones. But let's go ahead and do that too. Let's go ahead and copy and paste these two. Control C copy. Go into this folder. Control V paste. And then we're going to go to our studio. And we're just going to go back to the actually, yeah, because it doesn't have our full change. So we're going to put the, we can't just use the regular full file because we added the full change values to it. Okay, we'll chunk at the end right here. We could just, control it. Eval equals false. Write.csv data, all data FCs 36 to 42 by 19.csv row.names equal false. Let's run it. Okay, now we'll just cut this. It's not going to run again anyways because we put eval equals false, but after we add the let's actually write it before Control V, paste, great, Control S, save, good. Now we can close it. You don't need to save it, a short one. We'll go up a folder, it should be here. That's weird. It is here, it's right here. Control C, copy, go to the folder, Control V, paste. Now we can just cut it. My machine has enough room in it. If you're concerned about room, we can just cut this. We'll cut it. And then we'll take it up a folder to the R programs, all pathologies. And we'll paste it here. This is just for Tableau, so we'll add that to Tableau as well. We will be adding this one with the infinites and the nouns. Yeah. Okay, so almost 2.5 hours. Thanks so much. I think this was a good little analysis video. So next time we'll be looking at the gene summaries of these top 40 genes and then adding them to Tableau. Have a great night.